Good morning, thank you for the invitation. And before starting my lecture, I want to remember a great friend, a great scientist, Ferdinando Rossi, and he is responsible, as I am, of most of the work we did in the last few years on this subject, and unfortunately he died on January this year. But let's start with my talk. As you probably know, animals these are all rodents, and this a different type of animal that have similar size and similar metabolic rates, have a very different survival. And this reported 4 years for mouse and 5 years for raptors are indeed the maximum lifespan that has ever convincingly registered in these two species, and does not represent the normal lifespan of many strains of these two mice. And why I'm showing that? Well, I'm showing that because there is an obvious indication. If you have a maximum lifespan that is species specific, then even the cells that are making up the animal must have a maximum lifespan, or there is a mechanism that terminates somehow the life of the animal, and the cells can go on to live forever. This question is not a new question, and many of you are aware that most of the cells of our bodies have the ability to divide and continuously divide through the entire life of the animal. And there are high Flick limits to cell division, and many other studies trying to define if the potential is forever or stops after a while. But very few studies have looked at cells that lose completely their ability to reproduce, to divide, like neurons. Neurons are the quintessential cell in the body, and that once is differentiated, does not divide anymore. And the question, our question was, is the lifespan of the neuron similar or limited like the lifespan of the species where the neuron was generated? And in order to answer to this question, we took advantage, as I showed you before, of the lifespan difference into closely related species like mouse and rats. If you look at a different lifespan of mouse strain, you see that both the medium lifespan and the maximal lifespan varies quite a bit according to the strain, according to the genetic background. You are all aware that you can change quite a bit this survival curve, and then basically you can change the medium survival of the animal just by raising the animal in an axenic environment, or change the diet, and you can do many manipulations. But you can do very little to increase the maximum lifespan of the strain of this species. Most of the results are obtained by shifting the curve to the right and making a longer half-life for this new strain, but not touching the maximum lifespan. So the part of the brain that we were looking at is the cerebellum. The cerebellum has a very stereotyped structure. This is the molecular layer, this is internal granular layer, and those are the Purkinje cells, the main output neuron of the cerebellum, that is characterized by a very stereotyped dendritic arbor and very big shape of their cell body. So they are very easy to distinguish. They are completely generated before birth of the animal, and they do not divide anymore after they differentiate. And what we did was to show you the technique, was to make chimeras by injecting directly suspension of progenitors of Purkinje cells and other cells in the cerebellum into the embryonic, developing embryonic rat, taking these cells from a mouse. So taking these cells from a species that has a shorter maximum lifetime compared to the mouse one. And then we simply waited until the animal died and look if in mouse cells were still surviving in the rats. This is the scheme of the experiment. We of course had controls, and the controls were the siblings of the embryos that we used to obtain the neuronal precursor from the cerebellum. And if you sacrifice the animal just after the transplant and look at the still developing cerebellum, you see that the cells, the mouse cells into a developing rat cerebellum are distinguished by EGFP, because they come from a transgenic animal that express EGFP in all cells of the body, and they integrate with the rat cells and start to differentiate into a completely mature Purkinje cell. This is the same in a different animal, always at very early stages. And you can see that even other neurons in the cerebellum, like granular cells, are integrating and taking part into the cerebellar development of the host. And this is what you see in the adult. Those are granular cells, those are axons of the granular cells that make parallel fibers. And you can see that these cells are in the right position with the right shape, and they completely integrate into the host circuit. And these are the Purkinje cells. The green one, the red one are the host one, the rat one, and the green one are the cells derived from the transplant. Of course, we did controls, so we transplanted also developing Purkinje cells from rat into rat and from mouse into rat. And what you can see here is that there is a striking difference in the shape of the dendritic arbor, as expected, because the dendritic arbor and the cell body are slightly smaller in the mouse compared to the rat. So the mouse cells can integrate into the cerebellar circuit, but they retain their species-specific anatomy, and they can be distinguished not only by the expression of the EGFP protein, but also by their peculiar shape of the dendritic arbor that is simply shorter, taller, and do not reach the top of the molecular layer. Here is simply the statistic showing what you saw in the previous data. And here the survival curve in the experiment. So this is the survival of the siblings of the donor cells in the mouse, and this is the survival of the rats that received the transplant. And these red dots correspond to the days where we got the cells out of the cerebellum, and we checked if rat cells were still present. As you see, the half-life of mouse is approximately half of the half-life of the medium survival of the rats, and even the maximum survival under our condition that are normal access to food in a normal environment, in a clean but not carcinogenic environment, the maximal life survival of the two species is quite different. But as you see from these red dots, at all ages you can still find living neuron into the rat that are of mouse origin. So, for example, these neurons are actually surviving almost 2 times the half-life of the mouse. And this clearly indicates that there is no fixed time, or at least there is no a fixed time of survival for neurons, at least in this condition. These are just the picture showing you that you can find perfectly well the mouse origin Purkinje cell, perfectly integrated with the rat cells at all ages. And you remember that the median lifespan of our mice is about 18 months, and the maximum lifespan of these mice is 24 months. But you still find in the rat mouse cells after 36 months. But of course, neuronal aging, this is just another example. Do you have an increased number of Purkinje cells in the early stages of the transplant compared to the late stages? Well, this shows you that the number of Purkinje cells that you can find before 19 months is actually even less, probably for statistical reason, for random deviation from the norm, compared to the mouse that reach 36 months. So there is no indication that you have an increased death of the mouse neuron when they are put into a rat environment. But of course, this is not the only point that is linked to an aging neuron. Another very important factor of aging neurons is that they lose synapses. And by looking at the spines that are the postsynaptic terminals on the dendritic harbor in the Purkinje cell, you can count the number of spines into a 5 micrometer square, and do it many times, and have an idea that is very easy to obtain of the decrease of synapses. As you know, this that is linked to aging of the animal starts quite early in the mouse, and you almost have the loss of half of the synapses after 12 months, while in the rats starts much later. And what we saw is very easily seen by this diagram, showing that if you look at the number of spines into mouse, rats, and mouse cell transplanted to rats, you essentially have the same density of spine on the dendritic harbor when you sacrifice the animal at 1 month, and when you sacrifice the animal at 18 months, that is the medium lifespan of our strain. In the mouse, in the wild-type mouse, you have a very significant decrease in the number of spines, while in the mouse cells that comes from this same animal that are simply transplanted into rats, the number of spine is comparable to the spine of the host. And when you look at the maximum survival of the rats that have received the transplant, the number of spine is again, for mouse cells transplanted into the rats, is again similar to the number of spine in the host rats, and of course, because of aging, is also decreased. So, to finish, what are showing you this data, well, I don't know why, but well, what are showing you this data is that there is no maximum lifetime for neurons, and the loss of synapses is modulated by the environment. This, of course, is very good news for all people that is trying to increase the lifespan of a species, because we can expect that if you are able to increase the lifespan, there is no genetic limitation to the survival of the cell in our brain. So we are not going to generate a vegetative state patient by increasing the lifespan. And this is my message, and I'm ready to take questions.
This shows that the brain cells can live 1 or 2 more years than they were expected to. Do you have any indication as to an upper limit, as to if you had put these mouse cells into a a much longer lived animal, that they would still live then?
Okay, I think what you say is correct. This indicates that this mouse cells lives a couple of years more than the mouse cells that are left into a mouse. But you have to consider that the maximum lifespan of a mouse is 2 years. So I would read it as if you put in an environment that survives 2 times the normal lifespan of the animal, then you still have surviving neurons. So doing the experiment that you suggest is very interesting, but it takes very long time, because to obtain enough data with 3 years survival time, you have to do an experiment for 6-7 years. And if you want to increase by transplanting, for example, in a sheep or another species, you have to wait for 20 years to have the results. So I think it's a very good experiment, but very difficult to do in practice.
Paul will share this this morning, so he will take off and give us the...
Yes.
So have you tried to do the reverse, transplanting rat neurons into perpetuate to mice?
This is exactly what we are doing now, but of course there is a problem with that experiment, because the rat cells are supposed to live for 36 months, and the mouse as an animal is dying at around maximum lifetime around 21 months or 22. So you cannot do the exact opposite experiment, because the mouse is simply dying, so you don't know if the, if the rat cells are able to survive more. But what we are looking at that is even more interesting is if you put a rat cell that is into a mouse, is the rat cell going to lose the synapses earlier than it's supposed to be? And we still don't have an answer to that. But this is currently what we are trying to do. The opposite experiment that you are suggesting is very difficult. Is almost an impossible experiment, because the only way to do it is to transplant the entire mouse cerebellum with rat cell in a rat, and then it's a very difficult experiment. It's almost for theoretical reasons that are very clear, is almost impossible.
Do you expect to see the rat cells degenerate faster inside the mouse?
I expect to see that they are degenerating faster inside mouse, but of course this is my expectation, and I still don't have the answer.
How far is the experiment in terms of year? How long ago have you transplanted, and how long have these mice lived already?
Now,
Yes.
about a year, and you have to wait at least 2 years to have.
The results.
And then, of course, the success, the rate of success of the transplant, because you are doing them when they are in utero, you are doing in utero surgery. It's not 100%, so you have to generate many animals and then look for the transplant. So it is apparently a very simple experiment, but in practical, it's very difficult to do it.